I'd like to introduce Chris Schaffer, who will be talking about seeing the almost invisible using novel nonlinear optical effects for image contrast in biology and medicine. Chris.
Okay, good afternoon, everyone. My name is Chris Schaffer. I'm a faculty member in the Biomedical Engineering Department at Cornell University. And I'm also the current chair of the Biomedical Optics Division of the Optical Society of America. So today I wanted to talk to you a little bit about what I saw as sort of a key emerging area in biomedical optics that I think we'll see a lot of exciting developments in over the next few years. And indeed already some exciting developments. And this is going to focus on developing imaging tools and techniques that allow molecularly specific imaging deep inside tissue, deep inside scattering materials like brain or skin or other sorts of tissue. And it turns out that the best way to do this is through nonlinear optical methods, and I'll talk a little bit about why that's the case. And many of you are probably familiar with sort of the classic nonlinear optical imaging methods, namely two-photon excited fluorescence microscopy. So I'll talk a little bit about how that works today, but then I want to focus on some new developments in the last couple of years that have enabled new kinds of nonlinear optical methods to be used for generating image contrast. So I'm going to start by talking a little bit about imaging biological specimens, both thin and thick specimens, and the special challenges associated with imaging in thick specimens where optical scattering is important. I'll sort of advocate for nonlinear optical microscopy as one path. There are certainly others, but one path toward obtaining molecularly specific imaging deep inside scattering tissue with micron scale optical resolution. I'll then briefly talk a little bit about sort of the classic nonlinear optical contrast mechanisms. And a theme that'll come up repeatedly is how do we measure whether or not the nonlinear interaction occurred. So the idea of all these imaging is you're sort of mapping how much of a nonlinear optical interaction occurs as a function of space. And one question is, well, how are you going to measure it? And in all of the sort of classic nonlinear optical imaging methods, the way you measure it is the nonlinear optical interaction produces light of a drastically different color than what you send in. And so you're measuring against a null background, you're measuring the generation of new light. And these techniques include two-photon excited fluorescence microscopy, second and third harmonic generation, and coherent anti-Stokes Raman scattering, all of which have been used for ex vivo and in vivo, and in some cases clinical microscopy for biomedical research. I'm then going to spend a bit of time talking about some new ideas about how to measure nonlinear optical effects and form an image from them. And they're new in the sense that they're being applied to imaging, but for people who have done ultrafast spectroscopy in the past, you'll find that they sound very familiar. So these are tools and techniques that don't rely on this sort of large color change, but rather rely on other detection methods, namely modulation transfer and four-wave mixing, in order to generate a signal that can be measured. And in particular today I'll talk about degenerate four-wave mixing microscopy, stimulated Raman scattering, and nonlinear and excited state absorption measurements. So molecular or cellular specific imaging is critical in imaging of biological samples. And that's just because if you look at the image on the left, if you just take a picture of a cell looking at reflected light or transmitted light, lots of things look the same because cells are made of many of the same components. There's not large differences in refractive index. So it's nice to be able to have some tool that provides some kind of molecular specificity so that you know you're looking at one kind of structure versus another. So on the right is an image where different structures in the cell have been tagged with fluorescent molecules. In this case, the actin, which makes up the cytoskeleton of the cell, is tagged in green. the mitochondria, sort of the power plants of the cell in red, and the nucleus, the place where information is stored and genes are transcribed in blue. So with this labeling strategy, you can differentiate very fine structure in biological samples, in spite of the fact that without labeling, many things look alike. So one example that I'll come back to a couple of times would be, for example, if you wanted to image vasculature, one thing you could do would be to inject a fluorescent dye into the bloodstream. And then all the blood vessels and nothing else would be fluorescent. So if you took a fluorescence image, you'd be able to see those blood vessels, at least in theory. But in fact, what happens if you're imaging in a scattering tissue is because of the optical scattering, light that's emitted from the focal plane can scatter on the way back to the detector. And when it scatters, it's sort of like you lost track of where it came from. It blurs or it goes in other places of the image. So this brings up the background and decreases the signal, and you get a loss of image contrast. And in fact, if, at least in brain imaging, which is my area of expertise, if you inject fluorescent dye into the bloodstream of a rodent and shine light on it and image the fluorescence, you see just what this looks like, a big green blur. This is an example of this. The image on the left is a confocal fluorescence image of fluorescently labeled neurons in the hindbrain of a mouse. And the confocal image is one way to get around some aspects of scattering, and you can see that there's cell bodies and fine neural processes and a lot of structure there. On the right is an image that's just a wide field fluorescence image, and you can see all of that structure is washed out. And that's due to scattering of the fluorescence light on its way back to the detector, basically blurring, because of that scattering, you lose track of where it came from, you lose information. So if you could think, what would be the ideal way to be able to form a three-dimensional image of something, let's say a fluorescent molecule? What you'd really like to do is excite the fluorescent molecule or drive whatever mechanism is being used to generate contrast. You want to drive it in just one small three-dimensionally localized spot. So for example, with fluorescence, if you only produce fluorescence in one small three-dimensionally localized spot, then it doesn't matter how much it scatters on the way back to the detector. You're still going to know exactly where it came from because you only excited the fluorescence in one spot. So if we could develop tools that enabled the signal that we're using to generate contrast in an image to be produced in one small three-dimensionally localized volume and not be produced anywhere else in the sample, we could overcome a lot of the issues associated with imaging in thick samples where scattering tends to degrade resolution and mostly contrast. And it turns out that one of the best ways to do this is to use nonlinear optical effects for image contrast. So for example, the fluorescent molecule can be excited by a 2-photon absorption process instead of a 1-photon absorption process. So on the left you see a Jablonski diagram or energy diagram of a fluorescent molecule. That fluorescent molecule could absorb an ultraviolet or blue photon to promote it to the excited state, and then it would fluoresce. Or alternatively, it could absorb 2 infrared photons simultaneously. And the simultaneous addition of the energy of those 2 infrared photons will promote the electron from the ground state to the excited state, and then it'll also fluoresce. And if you think about the probability of that process happening, the probability will scale like the density of the fluorescent molecules, and then it will scale like the density of the photons squared, because you have to have 2 photons hit the same fluorescent molecule at the same time in order for them to collaboratively excite the molecule from the ground state to the excited state. So as everybody learns in kindergarten now, right, that photon density is proportional to optical intensity. So in the focal region, so the probability of this process will scale like the intensity squared. And so if you look for a tightly focused laser pulse, the intensity is very high right in the focal region and falls off rapidly as you move away laterally or along the beam direction. And in fact, so intensity squared falls off even quicker. So you can reach a regime where the only place where there's an appreciable probability of this two-photon excited fluorescence process occurring is right inside the focal volume.